Thanks so much, Chris. And yeah, it's really a pleasure to be here today to speak to you all about our initial experience with the Skope system on the Connectome 2.0. So the Connectome 2.0 is basically a project that we've been working on since 2017, 2018, when we put in the first proposal. And this is essentially the goal of this project was to develop a next generation connectome MRI scanner, really to deal with kind of some larger problems in neuroscience, especially in human neuroscience, of essentially being able to cover the entire brain for imaging, but then also getting information down to the microscopic level. And so this project, it was funded by the NIH Brain Initiative, and I'm excited to share some news about this at the end of the presentation about kind of opening up the scanner for users around the world to get hands-on with and also potentially to use our Skope system. So I think the fundamental kind of methods that we're advancing with the Connectome 2 scanner is really diffusion MRI. And so I would argue that diffusion MRI, amongst all of the MRI imaging contrasts, holds a great deal of promise for being able to get information down to the tissue level, down to that microscopic level. So we know that with MR, we are typically kind of limited in terms of our spatial resolution to millimeters in size, and the nominal kind of voxel size doesn't allow us to get really down to the microscopic level. So I would say that diffusion actually allows us to provide an indirect probe of structures down to this microscopic level. And so one of the ways that we do this is essentially if we know how water is basically diffusing through tissues and we have some sense of the geometry of those pores, we can actually start to infer information say about the sizes and density of cells and axons in the brain down to this micron level, which is basically smaller than we can actually see with our MRI measurements. And so I would argue that diffusion does hold a great deal of promise for allowing us to get down to sort of in vivo histologic information. Now, to get this kind of information, we need strong gradient systems. So why do we need strong gradients to get kind of microscopic information with diffusion? Well, on a conventional MRI scanner, in order to achieve strong diffusion weighting, essentially to sensitize your measurements to very small displacements by diffusion, we need to play out the diffusion encoding gradients for a long period of time. And this really kind of limits the minimum diffusion time and also pushes out the measurement to long echo times, which along the way diminishes signal to noise ratio simply by kind of T2 relaxation. So on the newest generation, and actually previous generations of high performance gradient systems with maximum gradient strengths going up to pretty much an order of magnitude of what we were able to achieve on previous kind of conventional scanners, we can actually make this process much more efficient. So what do these high performance gradient systems offer for diffusion microstructural imaging? Well, I think there's really three straightforward benefits. So the first is if you can do this diffusion encoding more efficiently, you can play out the amplitude more higher, then you can actually do the diffusion encoding much more quickly, bring in the echo time, and basically boost signal to noise ratio in that sense. More fundamentally, for diffusion microscopic imaging, the maximum gradient strength actually sets the resolution of the smallest structures that you can resolve with these measurements. And so higher gradient strengths allow us to improve the resolution of our diffusion microscope, if you will. And so I think this was a primary motivation for our efforts to go to the strongest possible gradients that we can, especially for human MRI scanners. And then the third benefit of these strong gradients is that you can achieve shorter effective diffusion times, and that allows you to really get a sharper definition of the diffusion probability density function and essentially resolve, say, shallower fiber crossings, more complex fiber configurations. And so this third reason was really the major driver for our original human connectome scanner, the one that we built through the Human Connectome project to really map all the major white matter fiber pathways in the brain. And so this was our original connectome MRI scanner. It's a 3 Tesla MRI scanner with a very strong whole body gradient built into the bore. This was installed in the MGH in 2011. We partnered with Siemens to build this scanner. And this scanner achieved a maximum gradient strength up to 300 millitesla per meter, which was the strongest gradient that had ever been built for a human MRI scanner at that point, about 7 times the clinical gradient strength. In addition to sort of the centerpiece gradient technology, you can see that it's actually whole body gradient. It was actually broken into 4 segments, each of the segments powered by its own separate set of amplifier for each axis. So you can see on the back end all of the gradient, kind of the power going into from the gradient power amplifier, those cables going into the gradient. So alongside the centerpiece technology, we also developed a slew of advanced acquisition technologies to get the most sensitivity out of whole brain diffusion MRI and to do it in clinically feasible time. So through the development of simultaneous multi-slice with Calvin Setzenpop and Larry Wald in our group, we were able to get whole brain coverage in much more efficient scan times. And then we also partnered with Boris Coil, who was in our Coil lab at the time, is now at Mittelhessen here in Germany. We had a very exquisitely sensitive 64-channel phased array brain head coil. So in aggregate, all of these advances allowed us to achieve nearly 10 times increase in sensitivity for high B-value diffusion. And we talk about high B-values, we talk about B-values on the order of at least 10,000, 20,000, going up to 40, 60,000. You can still see signal in the human brain, even at such strong B-values. So what did we use these gradients for? So as I mentioned previously, I think the explicit goal of the Human Connectome project was to map all the major white matter fiber pathways in the human brain. And we were able to do that successfully. We published and kind of publicly released a data set of 35 subjects worth of data with B-values going systematically up to 10,000. And that's been, I think, widely accessed. We've also used it for high resolution diffusion imaging, diffusion tractography. But I think one of the kind of important contributions that the Connectome instrument allowed us to actually demonstrate was that these dedicated high performance gradients can actually help us acquire a rich array of diffusion microscopic measurements that really get down to sort of the neural tissue microstructure. And so one of the key applications that we were able to demonstrate fairly early on was how these strong gradients could be used for axonal diameter mapping. So this was a paper that I had worked on early on with Jennifer McNabb, where we basically kind of stepped up systematically the maximum gradient strength on the scanner from the typical Prisma gradient strengths of around 80 all the way up to close to 300 millitesla per meter. And you can see that as you go to stronger gradients, the ability to resolve axonal diameters, essentially to sensitize the measurement to highly restrictive diffusion, basically diffusion within highly restrictive spaces like individual axons, which are on the order of microns in size, improves as you get to stronger gradients. And in fact, you need those very strong gradients to actually be sensitive to measures like axonal diameter. And so we basically kind of showed the promise of these strong gradients for probing tissue microstructure. Subsequently, we expanded this technique to mapping axonal diameters across fiber tracts in the whole brain. And so here I'm showing axonal diameter maps that we've averaged over 20 healthy individuals scanned on the Connectome scanner. And as you sort of average over multiple subjects, you can start to see that there's some really striking patterns of axonal diameter within the human brain. For example, that green triangle that you're seeing in the middle panel points at a very large fiber pathway. These are corresponding to fibers in the corticospinal tracts. These kind of are the large fibers projecting from primary motor cortex going all the way down into the brain stem. And so this actually is known to be one of some of the thickest axons in the brain. Agrees with what people have seen from neuroanatomical studies, also in non-human primates. Surprisingly, we were also able to kind of resolve some interesting patterns in axonal diameter across the whole brain. For example, this anterior to posterior gradient of smaller diameter fibers in the frontal lobes and sort of larger diameter fibers going into the occipital lobes. And this also agrees with studies that people have done in non-human primates and also in humans. I think there was a paper published by Leewald from MPI Biological, MPI of Biological Cybernetics here, where he actually looked at the human brain, kind of 4 human brain specimens, and showed that you actually do have different diameter axons in different portions of the brain. I think that these frontal fibers are finer to subserve kind of higher order processing functions, and we have larger fibers going back to the visual cortex, which is highly developed in humans. And so kind of armed with this tool, we were really interested in kind of developing the axonal diameter marker into a potential sort of useful imaging biomarker for looking at potentially axonal degeneration in the brain. And so one of the primary diseases that we focused on early on was axonal damage and multiple sclerosis. So multiple sclerosis is known to be a neuroinflammatory disease where you develop these sort of demyelinating plaques in the brain. There's relapses followed by remissions, but over time, people sort of, as they acquire more lesions, at some point they will actually stop developing these new lesions, but you'll see the brain start to shrink. And this is actually an ongoing process. It's believed that damage to the underlying axons by demyelination is the substrate for clinical disability that develops over kind of decades of MS in many patients, sometimes even more quickly if it's a very aggressive disease course. We know that the imaging correlates that are currently available for MS are non-specific for changes in axonal sort of structure. So even methods like diffusion tensor imaging are not entirely specific for axonal damage. People have detected decreases in NAA on MR spectroscopy, but that tends to happen later in disease course. So there's a need for improved biomarkers of sort of axonal integrity as the drugs for MS actually get better. So there's actually now an array of drugs that are out there to kind of prevent the neuroinflammatory damage. But we also are looking at kind of the development of new classes of drugs like remyelinating therapies that actually might be able to help protect the axons and kind of regrow the myelin around them. And so we need kind of better biomarkers for axonal integrity to figure out which patients would actually stand to benefit most from these sorts of neuroprotective therapies. And so in a fairly early study that we did, kind of subsequent to our initial studies of axonal diameter in the healthy corpus callosum, we imaged a series of MS patients who had lesions in the midline corpus callosum. And what we found even in this small series was that there was a systematic observed increase in axonal diameter corresponding to these demyelinating lesions and a decrease in the axonal density. And this finding was intriguing because it actually agrees with histological evidence of axonal dropout and swelling in MS lesions. So these are postmortem spinal cord specimens in a patient with MS and then a non-neurological control. And you can see in the MS specimen, this is the spinal cord kind of sample was stained with a silver stain to look at the axons. That the axons are somewhat enlarged, and there's also overall kind of sparser axons in this sample as compared to the normal control. And so it's not entirely clear what's driving these findings. These are basically postmortem specimens from people who have died after a lifetime of MS. There's various hypotheses as to, from the path literature, could it be that the axons are swollen? Is there some sort of preferential kind of dropout of smaller diameter axons? And then there's a compensatory swelling that occurs to try to preserve conduction velocities within these damaged areas of white matter. And so this really kind of highlights the need and the importance of having in vivo imaging biomarkers that get down to this histologic level, which is kind of how we typically diagnose disease, to try to bridge this gap and really bring it in vivo so that we could observe how the disease is sort of affecting people at different stages of their disease course. So we followed up this study in a larger cohort of MS patients, again, focusing on the corpus callosum, but now looking at not just the lesions, but also the normal appearing white matter. And what we found in this group of 40 MS patients, primarily with relapsing remitting MS, is a milder form of MS, or the sort of an earlier stage of MS, we saw that amongst all the imaging markers we studied, including DTI and brain volumetrics, that the corpus callosum axonal diameter was the strongest and most significant predictor of physical disability, as shown here on the left-hand side here, this is the EDSS, the Expanded Disability Status Scale. You can see that the EDSS scores really correlate with increases in axonal diameter. And what was even more intriguing was that the performance of the patients on various cognitive tests, including these two tests, the symbol digit modalities tests, this brief visuospatial memory test, were inversely correlated basically with declining performance as axons got larger. And these findings were particularly intriguing because cognitive impairment is a known kind of prominent feature of MS. Patients complain of sort of having difficulty remembering or thinking. And these functions are especially reliant on the corpus callosum for interhemispheric transfer. They're tests of processing speed, working memory. And so it's very interesting to see that amongst the whole battery of tests that we studied in these patients, that these two cognitive tests actually seem to pop out as being kind of the strongest correlates with axonal diameter. And so that brings me to sort of the motivation behind why we decided to pursue a next generation connectome MRI scanner. So why did we want to kind of go to stronger gradients? Why did we want to re-engineer the original connectome scanner? So I think fundamentally, we know that higher gradient strengths and slew rates are needed to achieve shorter effective diffusion times and also to probe smaller micron-sized structures. So one of the criticisms that we got in the early axonal diameter work was that these axons that we were estimating were larger than what we know most human axons are. So if you look at the error bars on kind of, and also just the scales on these graphs, you'll see that the axons are kind of estimated to be around 4 to 5 microns in size. And we know that most human axons are actually more on the order of 1 to 2 microns or smaller. And so what we and actually others have found is that these strong gradients sensitize us to some measure of axonal diameter, but really kind of more toward the larger diameter axons within this distribution in the human brain. And so this was one of the most, I think, important drivers of our effort to go to a next generation connectome. At the same time, we know that these diffusion measurements that we have are actually inferred quantities. So we're not looking at the axons directly. And again, this kind of speaks to another element of a scientific need that's been driven by our connectome efforts. So we know that we need better systematic kind of validation efforts looking at histology to inform essentially better biophysical models of brain tissue microstructure. And I think you've seen today from just the kind of brief talks that we got in the beginning, the brief hellos, that there's still a lot that can be understood about tissue microstructure in general, not just in the brain, but also in the body, for example, in the prostate, to really kind of better inform our understanding of the mapping between the diffusion MRI signal and what we're seeing in the tissue. And then finally, I think, and this is the grand vision that we have for these efforts, is that by pushing the limits of what we can do with diffusion MRI in the human brain and also in the body, it'll really allow us to infer how the brain's structure and its connectional anatomy are changing across scales with different types of behaviors, for example, in learning and memory. As you learn a task, for example, how to ride a bike, how is your brain changing? We know that structure predicts function in biology. And so basically getting a better look at how these processes are changing dynamically in a living human is extremely important. And then finally, actually to figure out how that structure is changing on pathology and potentially to develop earlier biomarkers of neurodegeneration, for example, in the brain, diseases like Alzheimer's disease we spoke about yesterday in our Meditech circle. Typical ways that you diagnose neurodegeneration in radiology are you look for tissue loss, tissue shrinkage. But by the time the tissue is lost, there's really nothing you can do. And so being able to identify these pathological processes earlier on, potentially at the tissue level, may allow us to intervene earlier and actually affect the disease course. So this is basically the Connectome 2 scanner. This was installed at the MGH last summer in July of 2023. And we were basically operational as of August 2023. You can see this picture is pretty new. This is when it was brand new, the plastic still even on the railings on the table. So this is a 3 Tesla scanner. It was built on the Vita platform that Siemens has. It's the wide-bore 3T, their latest generation wide-bore 3T scanner. And what we did was basically in the inner portion here, this is the gradient coil. And so we had to convert the gradient from a whole body gradient from the Connectome 1 design to a head-only gradient in order to achieve our sort of a target maximum gradient strength of 500 millitesla per meter. At the same time, we were also able to achieve a very high slew rate of 600 Tesla per meter per second. And this was really kind of aimed at being able to do more novel diffusion encoding paradigms, and also has benefits for image encoding. So we'll talk about this a little bit later. So in addition to the gradient coil, kind of building on following in the footsteps of the original Connectome scanner, we had an array of coils that were built for both in vivo human imaging and ex vivo whole brain imaging, kind of going toward that validation aim of our Connectome 2 project. So we have a very high 72 channel count head coil, kind of this next generation brain coil, compared to our original 64 channel head coil. These were both built by Boris Kyle. Then you can also see here, there's a 64 channel ex vivo whole brain coil. And that kind of builds on our original prototype of a 48 channel ex vivo whole brain coil. This is a bucket coil, essentially, that allows you to put the brain inside and then close on top. So integrated within these head coils, we have put the clip-on probes that have been basically very carefully positioned throughout both of these coils so that we can do concurrent field monitoring. And we recognized, I guess, from the early stages of the project that we really needed the field monitoring in order to be able to correct for eddy currents and other distortions that we anticipated with such strong gradients on the system. So to get a little bit more, give a little bit more detail on the Connectome 2 gradient coil. So as I mentioned, this is a head-only gradient coil now. It was actually designed, it's an asymmetric head gradient coil. It's designed with a 3-layer kind of geometry. It's actually built on the same geometry as the Siemens 7 Tesla impulse gradient coil, sort of piggybacking on the efforts that led to David Feinberg's first kind of impulse gradient. And that gradient goes up to 200 millitesla per meter with a very fast slew rate of 900 Tesla per meter per second. And that's built for 7 Tesla, very high spatial resolution fMRI. So this Connectome 2 gradient, it has this stepped design, which you can see with the 3 layers. The geometry of the intermediate layer allows you to put the shoulders of more kind of people within so that we can accommodate a wider range of patient sizes. And this gradient coil is now driven by 2 gradient power kind of amplifier sets to really get us up to this maximum gradient strength of 500 millitesla per meter and the slew of 600. And so in more kind of with more detail, these are the gradient specifications of the Connectome 2 scanner. Now that it's kind of been converted into a head-only gradient coil, the actual patient bore diameter is now reduced to 40 centimeters as compared to 56 centimeters from the Connectome 1. It has a very high efficiency. So because of going down to the smaller diameter, you can see that it's provided this boost in the gradient coil efficiency. And essentially, we're able to hit the maximum gradient strength and slew rates without issues. And as mentioned previously, the gradient power amplifiers have gotten basically better since the original Connectome scanner. So they are now able to provide about a third more power as compared to the original gradients, and they're actually more efficient. So we had to actually decrease the power, derate the power going to the scanner room to install this scanner. A little bit more about the gradient coil. So this was actually very cleverly designed, I think, to try to overcome some of the anticipated biological limits of going to very strong gradients. And this work was really spearheaded by Matthias Davids and Larry Wald in our group. So Matthias has built this really elegant kind of peripheral nerve stimulation framework. So we know that with strong gradients, stimulation of the peripheral nerves is going to be, was anticipated to be one of the major limitations of going up to our maximum gradient strength. And so we applied Matthias's peripheral nerve stimulation model together in concert with the gradient labs kind of design of the gradient coil windings to try to optimize the gradient coil design to minimize peripheral nerve stimulation. And so this figure really kind of illustrates the principle of the PNS optimization. So if you say started out with a gradient coil design like this, which is basically just has 2 layers, you can see that there's these nerve stimulation hotspots, essentially in the face right here, where you have more kind of angled and shallow peripheral nerves. And so the idea behind the peripheral nerve stimulation kind of optimization is that by adding, say, a mid layer to the gradient coil and adjusting some of those windings, you can push a lot of those peripheral nerve stimulation hotspots out of the face and down into the shoulders and even torso. And so the proof of this kind of optimization really was in the volunteer study that Siemens did on the connectome to gradient coil in the factory before they delivered it to us. So they basically measured peripheral nerve stimulation thresholds in 24 healthy volunteers. And these are kind of the data coming out of that volunteer study. So you can see with the connectome 2 gradient, that essentially the thresholds are now higher as compared to the connectome 1 across all different axes. And they really basically fall sort of at the upper limits of what the gradients are capable of performing. And so we have the gradient strength here on the y-axis and the rise times here on the x-axis, kind of demonstrating that we can indeed go up to close to 500 millitesla per meter with depending on the slew rate, and it sort of goes along these different axes. And I'll talk a little bit about our kind of initial experience with this in a little bit. In terms of the radio frequency coil, so these were also very elegantly designed by Boris Coil, who's now in Mittelhessen. And so he really tailored these radio frequency coils to have optimal form factors to get optimal sensitivity. And I do think that these coils really make the scanner. They complete the picture. As you can see, he did systematic comparisons of signal to noise ratio from the 72 channel coil to, say, a standard 32 channel in vivo coil. And you can see that there's a very high signal to noise ratio in the cortex and the periphery. And then also in the center of the brain, there's a boost as compared to the 32 channel coil. Similarly, for the ex vivo coil, he kind of did some comparisons here. He compared against the 72 channel in vivo coil sort of as an internal comparison. And for the ex vivo whole brain coil, you can see again that you gain some sensitivity, especially near the cortex, which was one of our targets for this type of validation work. So these are kind of images that we obtained in our first in vivo human brain diffusion MRI scan, which we performed on August 1st in 2023, soon after handoff of the scanner from Siemens. And we were very kind of encouraged to see that we could actually go systematically up to the maximum gradient strength. So here, sort of a typical B value at Gmax 150, then going up to 300, B of 4,000, and then finally to the B of 10,000 with a Gmax of 475 millitesla per meter and slew rate of close to 600. And you can see that even at the highest B value, you can sort of still see contrast within the white matter. I'll show you this sort of signal to noise ratio comparisons in a bit. But I think one of the most kind of reassuring things that we got out of this initial scan was that the patient or the participant who went into the scanner did not feel painful peripheral nerve stimulation, sort of evidence that we can actually make use of those aggressive target specifications that we had designed the scanner for in vivo human imaging. And so that was very gratifying to see. So we subsequently did a systematic signal to noise characterization of the connectome 2 gradient and the system performance, comparing against kind of equivalent measurements for a connectome 1 like protocol, and then also comparing down to a standard Prisma type 80 millitesla per meter type protocol. And you can see that as you get up to higher and higher B values, the connectome 2 does provide a visible improvement in the signal to noise ratio as compared to the connectome 1 protocols, also evidenced in the color FA maps. And these are sort of systematic plots that Hongxi Li worked on in our group, basically showing that as you go to higher B values, the connectome 2 scanner offers a reduction in TE by more than 2 times, especially at the highest B values, and that the improvement in signal to noise ratio is basically at least 2 times, depending on which B value you look at, compared to connectome 1. Now, when you compare to the clinical kind of gradient strengths, it's basically essentially infinite, even as you go to B values above 5,000 or so. And so what does this mean actually for the applications that we had really targeted the scanner for? Well, I think one of the key proof points for sort of validating our hypotheses about stronger gradients was in axonal diameter mapping measurements that we did in healthy volunteers. You can indeed actually resolve smaller diameter fibers with this with the connectome 2 gradient strengths as compared to the connectome 1. And so these are plots of axonal diameter in the whole brain performed on 10 healthy volunteers. We did a connectome 2 and then a connectome 1 protocol going up to Gmax of 600, 500 and 300 here. And you can see from the color maps that the axonal diameter now kind of goes down, the measurements can go down to about 2 microns or so. This is very much in line with what we had predicted prior to even designing and building the scanner. What I found was really actually interesting with these measurements was beyond the resolution improvement, the fact that the precision of the estimates is actually improved on the connectome 2 compared to the connectome 1. And so I think this actually speaks to the boost in sensitivity that you're getting. So better SNR allows you to improve your your estimation of these quantities of the axonal diameter. And this was also seen on the individual level. So if you look at maps of axonal diameter from, say, a single subject on connectome 2, you can start to resolve contrasts and detail that previously it was kind of hard to pick out on the connectome 1 measurements. We were very lucky on connectome 1 to have a few initial subjects who we were able to sort of see some interesting kind of contrasts on that gave us kind of the initial data to proceed with these measurements. But on connectome 2, it's actually so much more clear. And I'll also say that when you compare the connectome 2 maps in a single subject against kind of the maps that we needed to, say, average over 20 subjects to obtain with connectome 1, you can also see that this improved sensitivity will potentially allow us to improve microstructural detail down at the individual level. And so I think this really kind of speaks to this hypothesis we're now pursuing, is that we may be able to now map microstructure in individuals, kind of going toward this idea of precision neuroscience, that maybe we can actually extract information accurately at the individual level, because we know that everyone's brain is actually structured differently, they function differently. It's like there's these individual variations. So more to come on this. We've also kind of sort of used those strong gradients to demonstrate not just improved microstructure resolution, but improved spatial resolution for diffusion MRI. And so these are really nice kind of tractography results generated by Chiara Maffei and Gabriel Ramos-Jordan in our group, showing that with the connectome 2 gradient strengths, you can start to resolve fibers in very fine fiber tracts in the central brain, including this sort of medial tegmental tract, a medial lemniscus that previously was very difficult to see on even a connectome 1 protocol. And I'll note that these tractograms were actually created without any denoising on the scanner as compared to what we would need to do with connectome 1 to achieve even some measure of sensitivity for detection of such tracts. So finally, what are we doing now with the field camera? And so I think one of the primary things that we've been investigating all along with connectome 1 and now with connectome 2 is the effect of eddy currents on image encoding. So we observed on the original connectome scanner these very pronounced artifacts that we believed were basically created by eddy currents coming off of these strong gradients, essentially contaminating the image readout. This is particularly pronounced with these sort of multi-shot diffusion approaches, which we used primarily for ex vivo human brain imaging, one of the major applications of this scanner for validation. And so we invested in the dynamic field camera on connectome 1 to essentially allow us to measure the real-time phase perturbations, incorporate this into the image readout, and then get basically much higher fidelity measurements and image reconstruction with these highly segmented 3D EPI measurements in ex vivo whole brain specimens, as compared to without these accounting for this information, say with your standard one-dimensional linear phase correction approaches. These images that we obtain on the Connectome 1 scanner were usually contaminated by ghosting artifact, which would give a very strong directional bias in your FA maps, and basically gave rise to what one of our collaborators called the green problem with our ex vivo whole brain measurements. And so Gabrielle in our group has done a lot of work in trying to improve and basically measure the field trajectories on the Connectome 2 scanner. And so he's basically done this already for concurrent field monitoring using the 72-channel Coil, the built-in field probes for high spatial resolution in vivo diffusion MRI. And you can see from these plots that the K-space trajectories actually vary based on diffusion image kind of encoding, the diffusion gradient encoding directions. And so that would actually give rise to these ghosting artifacts that vary for different DWIs. And also variations in image resolution depending on the strength and directionality of your diffusion encoding gradients. And so he also showed, and this was work that he presented at the ISMRM, that the higher order kind of field terms are very prominent on the Connectome 2 scanner, indeed kind of justifying the use of the field camera for measurement of these kind of higher order field terms. And so here he plots the phase modulations between consecutive EPI echoes. They're highly nonlinear. They're not constant over time. And we actually believe some ways that these measurements will render the encoding not necessarily kind of linear time invariant. And so kind of speaking to the need for measuring these fields in real time. So here we've now kind of demonstrated that with the concurrent field monitoring, we can essentially recover higher fidelity diffusion weighted images at high spatial resolution. I believe these were done at 1 millimeter isotropic. So these images are coming off of the kind of reconstructed with the field monitoring data from the clip-on probes. These are results with just basically 1D linear phase correction with grappa. And then the dual polarity grappa on the scanner, it needed to be optimized. You could see initially that there was actually some noise amplification. We've actually since fixed in collaboration with Siemens, basically involved tweaking the regularization factor on that DPG kind of kernel. But yeah, I think we've been able to obtain very robust results with kind of high spatial resolution in vivo diffusion MRI using concurrent field monitoring. This is actually done at 0.9 millimeters isotropic with a b-value of 1200, t of 47 milliseconds, and this is the average DWI map. So we're now kind of tackling the problem of ex vivo diffusion MRI with whole brain. This was actually a hemisphere. It's even more challenging with whole brain specimens because you actually end up having to do very long acquisitions, larger matrix sizes. We are targeting a spatial resolution of actually 0.4 millimeters isotropic. This was done with 0.55 millimeters at a fairly modest b-value for ex vivo imaging, about b of 10,000. And you can see with just the online reconstruction that there's actually fairly pronounced ghosting artifact in the phase encoding direction. And so here we've basically started with dynamic field camera because it's an ex vivo human brain. We can actually do these measurements fairly precisely by putting the field camera in after our whole ex vivo brain acquisition. But then we sort of face some issues with kind of measurement of field trajectories on the dynamic field camera in the Connectome 2 scanner. And this was kind of work that was very recently kind of addressed and solved thanks to Cameron's help. So if you see here, these are the dynamic field camera trajectories that we are obtaining from these kind of using a field of view and slab encoding field of view equivalent to what we would use in the whole human brain. So you can see just the raw kind of FIDs coming off of the field camera, that they're actually starting at fairly low amplitudes and they were all sort of offset from each other. And then that basically leads to kind of loss of signal and unreliable kind of trajectories, which are also illustrated here. And so we were really puzzled by this. And these were actually very time-consuming and taxing experiments. So we had basically done the whole ex vivo whole brain acquisition for 36, 40 hours or so. And then we put the field camera in and we were getting these types of measurements. So thanks to actually Cameron's help and also systematic troubleshooting with Gabrielle, we figured out that there was actually leakage of transmit kind of signal coming from the scanner itself and basically tipping the field probes inadvertently. So Cameron was very astute and kind of observed this, that the 180-degree pulse within the sequence was not fully zeroed out when we were doing the field camera acquisitions. And this was actually kind of one of the challenges of dealing with kind of porting everything from an old scanner to a new scanner. We had to reprogram our whole 3D API sequence on the new scanner. And we realized that the ways that we were trying to zero out the voltages were not really completely taking care of the problem. So we actually had to zero out each of the pulses individually. And so you can see the pulse envelope of the 180-degree pulse here. So once we did that, we took out that transmitted signal, and now you can actually get really nice and coherent kind of FIDs. And so the trajectories now look very good. And so Gabrielle and another postdoc in our group is we actually just completed this acquisition. I think it's completing today because we set this up on Friday night. And I think the trajectories now look consistently great. And so what are we going to do with those trajectories? Well, there's actually several large scale projects that we're now kind of embarking on to make use of the Connectome 2 scanner for validation of connectional anatomy in the whole human brain, going all the way down to the axonal projection level. And this is basically a large scale effort. It's called the Large Scale Imaging of Neural Circuits effort, led by Anastasia Yandicki, who's a neuroscientist and electrical engineer in our center. And so she's basically put together this very elegant project that will essentially allow us to probe axons in certain areas of the brain. We're focusing on the internal capsule, going down to kind of the brain stem, using the Connectome 2 as a platform technology to do ex vivo whole brain tractography at very high spatial resolution. So we have two protocols, one that's targeting a slightly lower spatial resolution of 0.8 millimeters for microstructure, and then a very aggressive spatial resolution of 0.4 millimeters for whole brain tractography. And we will compare these and kind of merge this data with imaging from orthogonal imaging modalities, including optical imaging, light sheet microscopy, and then also with kind of whole brain hierarchical phase contrast CT to get microscopic level information about axons in the brain. And so lastly, I think we've actually been very fortunate to receive continuation funding for the Connectome 2 project, now in the form of this NIH Brain Initiative funded technology integration and dissemination resource grant for ultra high gradient MRI. And the goal of this grant is really to disseminate this technology by setting up the Connectome 2 scanner as a shared resource facility that will allow users from all around the world to basically propose and then get on the scanner to do measurements, say targeting microstructure or high spatial resolution, diffusion, tractography. And so we just received word on this funding about 2 weeks ago, and we're now setting up the kind of processes for this. So I really welcome anyone and everyone who's interested in using this scanner. I'm happy to talk to you all more about that. So I want to thank everybody from our center and also our local collaborators, local Siemens folks for all of their support of our system, and as well as the broader Connectome 2 team. This was really a multi-institutional collaborative effort making this possible, and I thank you all for your attention.